we will begin to look at some aspects of the central chemoreceptors. This has changed a great deal in the years since I started to study respiratory physiology. And we'll look at a little brief history of the background of it first. If we look back to the 60s and 70s, we see that what these folks did and published was that they turned the animal experiment upside down. Whoops, let me can we admit all? Okay, is everybody here? Nobody's waiting? Let me know. Yell at me if you're waiting. Sorry, if you see somebody waiting. Obviously, they can't yell. Anyway, what they did was they turned the animal upside down. So they were looking at the ventral surface. Not an easy thing to do because there's an awful lot of things in the way that have to be surgically removed before you can, including bone and lots of soft tissue, before you can get a look at the ventral aspect of the medulla. And the experiments they did were very crude, but they did tell you something. And that was to take tiny pieces of filter paper, soak them with high 109 concentration in acidic solution, and then place them on the medulla in various places. And Mitchell and Lushka himself, together, those two, looked at this area here, which is now being called the M or Mitchell area. And Lushka, of course, found a place down here. And what they found was that when they put these acid pledgets of filter paper on the surface, respiration increased. And so the conclusion was that they had somehow or other found where the central chemosensitivity areas were. And they were, for the most part, rather right, because indeed they are very close to the ventral surface. And I'll ask everybody to mute. I have somebody there. Now, I met Yasumatsu Okada at a conference, and I really enjoyed his slides and persuaded him to loan me this one. It is published back in 2001. And here's the experiment he did. Here's a slice of the brain. Let's see, it doesn't say whether it's from, I think it's probably from rat. And what he used was a voltage sensitive dye, which neurons took up. And then he would increase the CO2. And he would look at under the microscope and see what he could observe. And sure enough, the raphe obscurus and raphe pallidus, these two areas here, and along the ventral surface, all lit up when you gave CO2. And if you looked even closer, you could identify something that looked like where the ventral respiratory group would be. And sure enough, it lights up. And that illustrates the problem, because we're pretty sure the ventral respiratory group is not chemosensitive, and yet it lit up when the CO2 is there, because you have increased ventilation, and this is a transmission from the chemoreceptors, which are driving this, down to the spinal cord to see our output on the phrenic nerve. So how do we determine and eliminate these simple conductors of the stimulus down to the respiratory muscles and concentrate on the central chemoreceptors? Well, if you bathe the solution in something that blocks synaptic transmission, a tetrodotoxin or a cadmium, high cadmium to substitute for calcium, then you see the same experiment when you give CO2, you see these ventral areas light up. And if you look closely, you can see they seem to be grouped in what looks like little groups of neurons here. And so that is a very elegant demonstration of exactly where the central chemoreceptors are. And that was in 2001. Then Patrice Guillenet and his laboratory have been working on this problem, and they came out with this beautiful paper in 2019 where they summarized what they thought of the retrotrapezoid nucleus as the central chemoreceptor and the regulator of breathing automaticity. Because if you block it, breathing stops. In other words, that constant drive that we required in our respiratory rhythm models, that comes from the central chemoreceptors. He's quite thorough in this paper, and there are several sections there: sleep apnea, which we will get to later, and postnatal stuff, the developmental aspect of the respiratory physiology, and then the idea that there might be a parafacial expiratory oscillator. And that goes into one of our theories of rhythm generation, where people have suggested that it's coupled oscillators rather than sort of one big one. Not sure I agree with that. So, as he points out here, he's found the cellular and molecular basis for chemosensitivity, the types of cells that are involved, and their molecular identity, so that you can identify where they are. You can put viral tracers with opsins in them, so you can control them with light. And he's also provided a debate of the prevailing ideas with regard to location, and that is that there are diametrically opposing views. And the first one is the distributed chemosensitive theory. This has been championed and because of the experiments done by Jean Natty. And for a while, that was the major theory before the Patrice's lab came along. And so he suggests that the central chemoreflex results from hydrogen ion that is sensed in a single brain site at the ventral surface, and that is named the retrotrapezoid nucleus. Well, what about the idea from Jean Natty here, Eugene Natty? There is substantial evidence that many sites participate in central chemoreceptors, some located at a distance from the ventral medulla. Well, as we saw in Okada's experiments, the raphe, for example, lights up, and some people have thought that is definitely the case, that it is chemosensitive. But here we are, is my comment. How do you differentiate chemoreceptors from premotor neurons in these kinds of experiments? And that applies to this kind of experiment, which used microdialysis. So here's another methodology to consider. You put in these microvial probes. It has 20 microliters of brain extracellular fluid inside it, and that is circulated through through one and out the other. So it comes down and out. So it's the outside is exposed to a semi-permeable membrane so that molecules can diffuse out. This is perfect for putting an acid solution in here and circulating it so that you affect the local area and cause a local acidosis. And then you can see, did respiration increase? Did I have an effect? And sure enough, as he found, many sites can stimulate ventilation with a microdialysis probe. And that's what led him to propose this theory of his, the distributed chemoreceptors hypothesis. There's the problem, I think, that occurs with that. So it's difficult to make sure that you're actually stimulating chemoreceptors rather than just the neurons that transmit respiratory rhythm and the chemoreceptor drive. So now we come to Guillenet's paper, the central chemoreceptors and the retrotrapezoid nucleus. I put in quite a few little bits and pieces from his figures here that we should review in detail, and pointed out some of the salient points in red here of what we're looking at. And so this describes an experiment where we can use the lentivirus with opsins, channel rhodopsin, CHR2 abbreviation, which you can then stimulate and pulse with laser light. And you can record with the microelectrode a unit while you pulse it. You can also record the phrenic nerve discharge and see how that occurs. And where are these neurons? Well, you can stain them. B is an example of the neurons that express the channel rhodopsin. Takes 3 weeks for it to take. And they're located in the marginal layer. They also have nuclei that exhibit FOX2B. And for those that understand the genetic implications of how these labelings work, which I do not do well, that does characterize these particular neurons. And he sees one right here in green. And as Okada saw, right along the ventral surface, we have these neurons that picked up the channel rhodopsin. And so here we can turn the laser on and see what happens. And the discharge in bursts per minute is here in the unit that we're recording. And here is the phrenic nerve discharge. And so you can see that, yes, we recorded the phrenic nerve, and certainly turn the laser on, you get a stimulation. And here's, oh, sorry, this is the phrenic nerve discharge. This is the unit. And this is the unit. If you ventilate so that you lower the CO2 sufficiently, you can turn off the respiratory rhythm and simply turn it on again by activating the retrotrapezoid nucleus. That tells you that that is where the drive to the respiratory rhythm generator is coming from, that keeps it oscillating. If it's absent, it doesn't oscillate. You don't breathe. And here's a case now where they can get and record from the neurons and see what happens to phrenic nerve discharge as well. So this is pretty convincing information. So we'll see that it's also drives breathing in proportional to arterial pH, or arterial hydrogen ion concentration, as I would prefer, but there we are. So the kinds of experiments, there's where we are. We can stimulate in the ventral respiratory group and record down here, and we can look at the neuron discharge of this retrotrapezoid neuron, and we can change the CO2, we can put in hypoxia and kynurenic acid and so forth to block, and we see responses. And we can see that if we change and look at arterial pH here, we have a linear relationship with the retrotrapezoid neuron, the firing frequency here. And you'll notice that when we get down below this, or below hydrogen ion concentration, above pH threshold, it stops. So, point one, the central chemoreceptors' response to CO2 has a threshold. We will see this echoed in ventilation when we study that aspect of it. They also respond to hypoxia via a polysynaptic input from the carotid bodies. The drives seems to add on to what the retrotrapezoid neurons are doing to stimulate the ventral respiratory group. So here's the laser experiment here, whereby we've turned the laser on, and we've certainly got an increase in flow. So, and this is the difference here, is that we have unanesthetized rats, so we've decerebrated them. So we can optogenetic activation of RTN neurons, strong increase in breathing. It's comparable to that which is evoked by hypercapnia. And as they say, it's mediated by the release of glutamate, because if you delete the glutamate transporter, then you don't get so much of a response. So that's telling you how this system works, how these neurons work. But I have my own ideas about that too. So there we are. Here you can instantly silence them. So you can see where they are. The green laser reads your breathing rate. You can see the frequency and the flow of breathing here. And if you get hypoxic, then this elimination here, you can't do it. So this gives an idea. So that, first of all, the effect on breathing, when you silence them, the firing rate is, in anesthetized animals, the proportional to the pH threshold, again, figure 2C in there. And these characteristics are consistent with what would be expected. And in some, with or without anesthesia, the lung ventilation is proportional to the mean level of activity of the RTN neurons, and it has a threshold. Where are they? More details here. This is a Sprague-Dawley rat, and they identify it by immunoreactivity FOX2B and the absence of tyrosine hydroxylase, left side. So there's various things in here in the way that you can stain and identify neurons that will probably be better understood by some of you than I. But here is the surface of the ventrolateral medulla. Here is a neuron, and you can see tracing out its dendrites and axons, and it has axons that go down all over the place into probably the ventral respiratory group, and that is going to be where they are located in a series of slices that are displayed transparently here. So they are in a little column here. And that was done in the mouse, in G there. And what else do we say? The dendritic structure of two representative neurons in D, and that's in anesthetized rat and stained by biotinamide. And that's an intracellular stain. And there we are. Within 200 millimeters, micro, 200, I think, that I don't think that's correct, because they're extremely close. Look at the here, we're 0.1 millimeters, very, very close. And the RTN neurons may therefore be sensitive to what the hydrogen ion concentration is in the cerebrospinal fluid, which is bathing the ventral surface. Something else to consider, not just coming in via the blood to the brain. So here's a summary. The evidence that RTN neurons, dry breathing, and proportional to arterial pH have a threshold below which they are silent. I keep belaboring this point and putting up these slides here to prove it. So how might they also work? Well, a lot of people like Alexander Gurin and company, along with Mike Spire, Sergei Kasparov, they thought that astrocytes may play a role. And this is to remind you that there's a complicated slide in their paper that shows that they can evoke calcium responses in the ventrolateral area of the brainstem from these astrocytes. Why look at calcium? It's because they are not excitable, but they do display calcium excitability. Therefore, they may release substances which activate RTN neurons. So they said, yes, acidification induces rapid increase in calcium, and show the various experiments by looking at the various stains that can be done to light them up in these pictures, and looking at the fluorescence versus time and what's happening with stimulation as well in these diagrams here. So that is something else to bear in mind, and we'll see why in a moment. Because we're going to hypothesize what the detection mechanisms are. This was again summarized by this paper of 2019 from Guillenet's lab. So what are they? In A, we illustrate what they are. And number one is that they are simply sensitive to hydrogen ion concentration. That pathway one. It could be paracrine activation, that's by ATP? No. Rats. I can't remember. But something released by astrocytes. And then inputs from neurons themselves, perhaps, which would be 3. So other neurons nearby might cause them to discharge. And changes in the blood vessels in the area, if they constrict and therefore do not carry away the CO2 that's produced in that area, CO2 will increase. And that could affect the discharge of the RTN neuron. But eventually, whatever the method, they fire and they provide an excitatory input to the respiratory pattern generator. So here's a look at what kind of thing might be happening for the sensing of the extracellular hydrogen ion concentration and causing it to increase its firing pattern. And this could be caused by raised CO2 or lowered pH. I'm going to propose that it is a cell autonomous sensitivity, but not to the extracellular. Then we can look at C, which has the astrocytes release. There we are, ATP. That's paracrine. That's what I was trying to think of. That releases that and therefore stimulates the RTN neuron to activity to the respiratory pattern generator. And this 2019 paper, this is their sort of summary of how it all might be working. Inputs from the spinal cord, perhaps from raphe, which we do know raphe neurons do respond to CO2. Locus coeruleus, maybe. Other things in hypothalamus. Perhaps these come in as well as the innate sensitivity of the retrotrapezoid neurons. The carotid bodies, of course, relayed via the nucleus tractus solitarius and the dorsal respiratory group, and stretch receptors from the lungs also come into play perhaps as well. And they will look at this again towards the end because they have given a sort of revised version of this. Well, the other aspect that is very important here, the complementary control of breathing by the RTN and the carotid bodies. And so they provided this statement, which gave me to understand that the peripheral and central chemoreceptors drive to breathe are additive. And indeed, if we look at them in humans, we'll see that they are, even though there are disputes about that. So in sum, they provide two sources of drive, carotid bodies and RTN. They contribute unequally because the carotid body drive depends on the level of oxygen. They are highly sensitive to hydrogen ions in hypoxia, but not so sensitive when the PO2 is high. And so when breathing is compromised and PO2 decreases, you get more carotid body and PCO2. They rise concurrently. And that's why if you try and strangle somebody or you try and hold your breath, then both the carotid bodies and the central chemoreceptors are going to force you to breathe. Recruited in parallel. So that's an important point to remember, as we will see when we get to the human experiments. So here's my suggestion for an intrinsic sensitivity to CO2. And it comes from a model that we will actually look at. It's the publication. We'll look at this paper for next week and the peripheral chemoreceptors. But I made it up to understand smooth muscle vasoconstriction and vasodilation, which controls cerebral blood flow, because they are virtually proportional to the intracellular hydrogen ion concentration. I think that the RTN cells may themselves be similar in the way that they operate. And so here's the idea intracellularly. CO2, of course, as we've known from the Henderson-Hasselbalch dissociation, we get hydrogen ion concentrations rising when CO2 rises. We also know from smooth muscle cells, but we don't know necessarily from the retrotrapezoid neurons, that causes calcium to rise. That increases the tone of a smooth muscle cell. And the theory that I propose is that the cell has an innate regulator of its own intracellular hydrogen ion concentration. It must do in order for the cell mechanisms and manufactories of various substances, proteins, etc., in order to work. And so in order to control that, it must control the hydrogen ion inside. The way to do that, if you cannot get away from the fact CO2 has done the increase, is to therefore do something about the strongly dissociated ions, which will therefore try and restore hydrogen ion concentration. So here's the normal hydrogen ion concentration that we want, and here's what we actually have. That sends a feedback to an error signal and a controller which operates to move strong ions in or out of the cells in order to try and control the hydrogen ion concentration. And this system, of course, is disrupted by CO2. It's also disrupted by hypoxia because you need energy from oxygen and ADP to ATP in order to make these transfers of ions across membranes. And so that may be the way that the retrotrapezoid neurons work. But that's merely my theory. Who knows? Maybe one of you will take on the task of finding out whether it's true or not. So the final words from Patrice Guillenet here are that acid detection is an intrinsic property of the principal neurons. And according to them, it's enhanced by the paracrine influences from the surrounding astrocytes. It also depends on the vascular constriction, perhaps, as we saw. And so it's the RTN that mediates the hypercapnic ventilatory response via projections to the respiratory pattern generator that we looked at last week. That's from his 2022 paper, so we're quite up to date. And here his revision of that signal, that figure that looked at everything. And so that's very nice indeed. If you are interested, that article also discusses the sympathetic and parasympathetic control from the rostral, ventrolateral medulla, because that's where the sympathetic outflow and parasympathetic outflow neurons that generate those signals that operate throughout the body, that's where they come from. But that's not particularly respiratory, so we've ignored that for now. So what about some other aspects of the central chemoreceptors? Well, they are in the intracellular fluid surrounding neurons in the central chemoreceptors. There's interstitial fluid, there's vascular smooth muscle between the cerebral blood. So cerebral blood flow certainly controls CO2, and that equilibrates. CO2 goes everywhere, driven by its partial pressure gradient. The ions don't, they require transport and so forth. So the point of this illustration is that the cerebral hydrogen ion concentration surrounding the environment of the central chemoreceptor may differ from blood hydrogen ion concentration that one might measure during an acid-base disturbance. So you cannot assume that the central chemoreceptors are seeing what you see in the blood necessarily. Now that has another aspect to it. But before we get to there, let's convince you that indeed cerebral blood flow changes with CO2. And here we have a rebreathing experiments that were done, and we're looking at a end-tidal PCO2 trace from a human subject. There they are at rest. There they hyperventilate at first for a few minutes, 2 or 3 minutes, and then they begin a rebreathing experiment. This is not the way I do rebreathing. This is not quite right, but it does illustrate the fact that, yes, if you do a rebreathing experiment, you get a slow rise in CO2 over time, simply because you have cut off the elimination of CO2 via the lungs. You're just breathing it back in from a rebreathing bag. Therefore, the only source is metabolic CO2 being produced in the tissues, and that accumulates rather slowly at your metabolic rate. And that is also determined by how big the bag is, how big the lungs are, and the size of the CO2 compartment in the body, which is quite large. So it rises rather slowly. And that gives you time to take a look at things like the sympathetic nerve activity. And you'll see that this goes along here as CO2 begins to rise and meets the threshold and suddenly begins to increase and looks like it increases more and more and more. Here's heart rate, doesn't respond so much, responds while they were hyperventilating, as it does, but then settles down, does rise slightly. But the arterial blood pressure certainly rises. And the cerebral blood velocity, in other words, blood flow, measured by a transcranial Doppler, also rises. So there we have the fact that if you measure transcranial Doppler of the middle cerebral artery, the blood flow goes up. The blood pressure also goes up. That's going to increase cerebral blood flow. That's one of the things that happens. The smooth muscles of the blood vessels controlling blood flow to the brain vasodilate with CO2 as well. So both aspects, pressure increase and vasodilation, cause this increase in the middle cerebral artery blood velocity or blood flow with CO2. And we have the sympathetic nerve being activated as well. It's becoming more and more important when we consider the carotid bodies next week and the aspect of what they do for high blood pressure.